A-Level Biology 9700, Paper 5, Variant 2, May June 2024. Question number 1. So, question number 1 is tree plantations are areas where trees are planted for a particular purpose. Some tree plantations increase the supply of wood for construction and fuel. In many plantations, fast-growing alien tree species are planted. Undergrowth is found in and around plantations, increasing plant diversity. Underground is mainly made up of indigenous species, small plants and shrubs. Some parts of the plantations of eucalyptus trees, and you can see Bangladesh has many tree plantations as part of a national tree planting program. Two fast-growing alien tree species planted in Bangladesh are Acacia auriculiformis, and Eucalyptus calameldensis. Acacia and Eucalyptus are considered to be invasive species as they outcompete indigenous tree species such as the salt tree, Shoria robusta, and Mango uh, magnifera indica. Scientists carried out an investigation into biodiversity of plant species in the undergrowth in plantations of alien trees compared with the undergrowth in plantations of indigenous trees. The hypothesis that the scientists tested was stated as the undergrowth in plantations of alien tree species will have a lower biodiversity. Undergrowth in plantations of alien trees, they have a lower biodiversity of plant species than the underground in plantations of indigenous tree species. So this is the hypothesis which was made by the scientists. So identify the independent variable in this investigation. And students, this we according to this hypothesis, we have to observe the biodiversity in the tree species. So the independent variable can be the species of trees. In this case, the independent variable species of tree, or you can say the tree species or the types of trees. So for the investigation, plantations were selected within the same region of Bangladesh with the same environmental conditions. Tree plantations of each tree species were selected: acacia, eucalyptus, and salt tree and mango. A plot measuring 36 meter by 36 meter was studied with each plantation. For each plot, belt transects were used to collect the data needed to determine Simpson's index of diversity. For each type of plantation, Simpson's index of diversity was calculated for the plant species in the undergrowth. Each plot was studied in April, July, and November. Uh, Now, for the each tree of plantation, it studied in April, July, and November. Identify two variables the scientists standardized in this investigation. The two, you can say, the variables which should be standardized in this investigation, the different plant, three different types of plant species will be observed or will be used for this investigation. So if we are talking about the different species of the plants or so there should be the one variable which should be standardized in this case, that the number of plants should be the same. That is, the same number of plants of each species should be used for this purpose, or in other words, same number of plants should be used for this investigation. all species or the three species which are used for this investigation. So same number should be used, and the same size of all plants should be selected. Same size, you can say same size, and the same number of plants uh, should be used for this investigation. So in this way, we can make a standard for this investigation. Now we are moving towards the next question, and next question is there. The scientists used bell transit to investigate the undergrowth of each plot. Describe how you could use bell transects to collect the data needed to determine Simpson's index of diversity of the undergrowth in each of the plots. Your method should be set out in a logical order and be detailed enough to let another person follow it. Students, you had discussed about the bell transect method in the chapter. Uh, so here, first of all, I would like to, uh, and this question is of 5 marks. So 5 marks we have to write about uh, the points, uh, which will help to gain you the 5 marks. So first of all, I would like to tell you what is the quadrate or bell transect. So quadrate or bell transect is a method which is used to measure the uh, when there is a, you can say, change in the habitat growth, or you can say on one side there is on one side of the plot or the one side of the field there are the different species are growing over there. So due to some reasons all the vegetations are greater in number, so we randomly we want to know about the different species of the plants which are growing at a place, or their number, and the number on one side is higher or lower, or you can say we can make comparisons by using this method. So quadrate bisect method can be used. There is a you can say proper procedure to use this quadrate belt transit. So for this purpose, we have to use this in this way. That is, we have to a measuring tape should be used for this purpose. One side of the plot to the other. So the 36-meter by 36-meter plot is used for this purpose to grow the different or the three different species of the plants for this purpose. So for the miring tape should be used for one side of the plot to the other, and then we have to put a quadrate uh, on the start of the tape, and then we will count the number or estimate percentage which will be covered by this method, and then a key can be used for this purpose to identify the different species, and then the results will be recorded in the form of a table. This is the proper method which we can adopt. So then the quadrate can be moved along the tape, and then from one you can say in this way, this is a tape and quadrate, it will be used in this way. So randomly the quadrate will be used for this purpose, and from one side to the other it will be moved on. So randomly it will be used at different points of the plots. So repeat this method will be repeated again and again, and the quadrate will be used again and again to collect or calculate or to count the different species over there. So continue until the full length of the miring tape have been sampled, and then calculate the average of each species, and in this way we can draw a bar chart. We can draw a bar chart to know the different species which are going over there. So it was all about the quadrate ball transect and the method which we can adopt to investigate. So now I am going to write all these points which I have uh, told you. Now, first of all, we will make use a tape, measuring tape. You can ignore these words. So make use of measuring tape. I have explained the whole process. Now I'm writing these points shortly. They are made up of wood or you can say with any metal. They are available in you can say different sizes. So we will keep this quadrant along the and it will be first of all it will be kept at the start of the tape, or you can 0 meter of tape. Many samples can be taken after regular distance. So the distance which you do, it should be the equal. That is the after particular distance you can keep the quadrant to find out the or to calculate the different uh, variety of trees which are growing over there. So after this method can be repeated for many times. Then identify by the help of dichotomous key to uh, key the plant species. Plant species growing there. Count them, record them in the form of table, and draw a graph to compare them. So same method will be adopted for all species. So in this way, the process or the investigation can be conducted. Now we are moving towards the next part of the question, and that is Table 1.1 shows the Simpson's index of diversity values the scientists obtained for the different plantations during April, July, and November. You can see over there. The months for different months, the mean values for acacia is 0, 0.88, 0.85, 0.94, 0.88. So almost they are overlapping, and standard deviation is 0.02 for acacia, the same for eucalyptus, and the same for mango. Only the child has a little bit, uh, you can say less than them. So state and explain. The question is state and explain uh, whether the data in table 1.1 support the hypothesis. We are moving a little bit onward. That is the in this way to write on the given space. So the undergrowth in plantations of alien tree species will have a lower biodiversity of the plant species than the undergrowth in plantations of indigenous tree species. So these results do not support this hypothesis. Why? Because you can see the acacia and the alien species mango, all they have the same results. They have the same standard deviation. They have the same means. So it means the it the result does not support this hypothesis. So we will write this. Acacia and mango have the same values of means and S values. Now we are moving towards the next question. We had to write only one part for this purpose. Okay. The scientists prepared an extract of the roots of uh, staghorn sumac. Rose typus and shorthorn sumac is an invasive plant species. The scientists prepared a stock solution of the root extract with a concentration of 10 milligram cubic centimeter. The scientists then used the stock solution to prepare solutions with different concentrations of root extract. Describe how the scientists could use the stock solution to prepare 50 centimeter of solutions of solutions of the root extract with concentrations of 2.5 milligram centimeter square. So for this purpose, and 7.5 milligram cubic centimeter. So here you can see we will write the C1 is the stock solution, and C2 is 2.5 milligram cubic centimeter, and then volume V2 is 50 centimeter cube. So we have to uh, find out the V1. So we will use this formula. V1 is equal to C2 multiplied by V2 divided by C1. So first of all, for the value 2.1, we will use the value 2.5. 2.5 multiplied by 50 divided by 10. So the answer will be, if we calculate this, let me calculate. It will be in this way: 2.5 multiplied by 50 divided by 10. So 12.5, and the same will be for the V1 is C2 into V2 over C1 is equal to 7.5 into 50 over 10 is equal to, so I will also calculate it: 7.5 multiplied by 50 divided by 10, so it will be 37.5. So it means we need the concentration, for concentration 2.5. 2.5, this is for 2.5, this is 7.5. We need volume of water, this volume of water, volume of water of water. Okay, and then for what will be the volume of stock? For 2.5, the volume of stock will be used as 12.5. This is 7.5, and 12.5 and 7.5. This is 2.5, and this is 12.5, and this is 37.5. So the volume of water will be, which will be used for them will be in this way, and then the volume of stock, it will be used this, and then the volume of distilled water. For 2.5, we can make a table for this purpose in this way. I think we should make a table over here, which will give us the proper results. The table can be made over in this way. 2.5, first of all 7.5, then 2.5, and then 37.5, and 12.5, and 12.5, and 37.5. So this is the concentration. So volume of stock, and the volume of water. So in this way, you can calculate the answer. So this is the volume of stock, and this is the, we will add the volume of water. I hope you have understood this. We are moving towards the next question. And the next question is, in some regions of China, marigold plants, targets erecta, are grown commercially. In the investigation into the effect of short stage for sumac root extract on the growth of marigolds. The scientists carried out the procedure described in step 1 to step 5. Prepare 5 containers, each with 5 marigold plants grown from seed. Add 20 centimeter cube of distilled water to one of the containers. This is the control. So control, add 20 cubic centimeter of the root extract concentrations to the other 4 containers so that each container has a different concentration of root extract. Repeat step 2 and step 3 each day for 60 days. After 60 days, remove the marigold plant from the soil and untangle the root. Measure the length of the longest root for each plant. The scientists replicated this procedure twice and calculated the mean maximum root length for the control and for the root extract concentrations. The results are shown in table 1.2. So you can see the control value. It is 163.0, and for 2.55, 7.5, and 10. So the different values has been shown. The maximum mean root length after 60 days are different. In control, it was 163 millimeter after 60 days, but in the at other it is 10, 141. It is decreasing. So with the you can say the root extract concentration is increasing. The mean maximum root length is decreasing. So using the data in table 1.2, calculate the percentage changes in the mean maximum root length between the control and the root treated with 10 milligrams cubic centimeter root extract concentration. Show your working and record your answer to 3 significant figures. So we will take these values and we have to find out the answer in the percentage. We are finding have to find the percentage change. So it is between the concentrate root extract concentration T, and we will find out in this way. The formula, I'm going to write the formula first, and we will put the values into percentage change is equal to new value, or we can say minus initial value, or you can say the final value or the different. So I have written the new value and the old value with the into 100. So here we are going to write, and you can show the it is decreasing. So the percentage change is towards the decrease. So we will take 141.6 minus 163.0 divided by 163.0 and multiplied by 100. So after, let me calculate this, and after calculation, the value is in this way, minus 21.4 divided by 163. So the answer is minus 13.1%. Student, it takes time when I do all my work in front of you, but it will help you to understand. So for this purpose, I always do it in front of you. So and it will be divided by 163.0. So this is 13 point 13.1% decrease, or you can say the percentage change is 13.1. 13 point. Next question. A student suggested that this investigation could be improved with reference to the method used and the data in table 1.2 state and explains the three ways to improve results. Uh, okay, so we have to write 3 points for this purpose that which are able to improve this method. So you can say more concentration values can be used. There were only few, so more number of these values can be used. So if there are to be studied, then exclude the anomalous results. So it will also help us to reach up to the correct, uh, you can say accurate results. And repetition So in this way, the we can be able to get the accurate results. Next question is to investigate the effect of the different root extract concentrations on the diversity of microorganisms in the soil. The scientists took samples of soil from each of the containers, identified and counted the number of species of microorganisms present in the samples, calculated the Simpson's index of diversity values for the species of microorganisms in each of the soil samples, state two variables the scientists should have standardized when taking the soil samples. Two standardized soil samples. So two variables we have to talk about. The two variables. So when the scientist took the samples, they should have to take the same mass of the soil for this investigation. And secondly, same type of soil. Same type of soil. Soil and use it at the same time, because there can be the change in the you can say soil type due to some reasons, due to water, due to addition of water, due to you can say less water, due to introduction of some insects into it, or soil insects into it, or bacteria. So in this way there should be the same type of soil should be used for this purpose, and same mass of the soil should be used at the same time. So it was all about question number 1. Students, please like, share, and subscribe my channel, and thanks for watching.